You know, as a segue into this, I ran a billion-dollar enterprise in North America in diagnostics. And it's not easy convincing companies to go into these avenues. And I just want to say with the malaria project, it took me 5 years and an intervention from a friend to make this happen. So, just click on it. Yeah. So this instrument here is the Muse, and it's been out now for about 8 years. It was first introduced as a research unit. It has all the cell analysis tests on it: cell pathways, cell signaling, cell growth, cell proliferation, all of the other essential tests for cell analysis on it. Initially, it was designed for the laboratory of the developed world, and I took it to Cameroon, and it had 19 parts on it, and I replaced 17 to accommodate for humidity and heat that you see in the tropics. So I lived through some of the pains the gentleman was talking about earlier. We have 3 tests that have diagnostic capability on them. The first is a cell viability test that allows you to know if you're looking at cells for research that they're alive and active. The second test on this has been for HIV monitoring, and it's a CD4, CD8 test, and it's the most sensitive T-cell enumeration available on the market today. Because 40% of the AIDS patients die of malaria in sub-Saharan Africa, I was convinced, and I worked with the company to develop these malaria assays. The first one is a bead assay, and essentially you take an antibody coupled on a bead with a target, you add the blood to it, and you conjugate it, and then you run it on the system and gets results. It's a simple test, 2 dilutions. I tell anybody that if you can operate a push-button screen and pipette, you can do anything on a Muse. The second test I'll talk about is cellular detection. It does the parasitemia percentage and the parasite density. And this is great for testing drugs. So the way it works is we have markers, HRP2 and LDH on the beads. The markers fluoresce and can be identified as they run with the blood sample into the flow cytometer. Flow cytometer has an endless data capacity. I tell customers, if you can fill up it, we'll give you a free one. So you look at the protein markers, you add the sample, and you get the results. So the reason we use HRP2 is to eliminate the false negatives that sometimes go with technologies that only rely on LDH as a marker, particularly for Falciparum. And it's simple to read. A negative, everything's shifted to the left. If I have a Falciparum, I get HRP2 and Falciparum LDH. And with Vivax, I get a Vivax LDH. So very simple to read. So the first thing we did is we tested it on standards that we got from FIND. And we did this in-house because it's always hard to get blood samples, especially fresh blood samples, with malaria when your lab is in California. And we basically hit it on every one of the standards that they gave us. We even diluted this sample to see how far we could go on different levels, and we hit everything. Oop, let me go back. Okay, that's where it was. Okay. So next we went to blood samples, and these were frozen samples that we got from friends in Uganda. And we ran adults and pediatrics to find out in the lab if it would work with both. And we went in everywhere comparing results from 24 parasites all the way up to 120,000. So we knew we could do the range. So next we evaluated samples from asymptomatic pregnant women. And this was a collaborative study with UC San Francisco. Somebody was talking to me about UCSF earlier today. We ran 30 samples, and we compared them to microscopy and also PCR. And we had a high sensitivity down to 1 parasite per microliter. Then we decided to do Vivax, and we got samples from Dr. Thu in Vietnam, and we ran them on the system and went down to 79 parasites, but we now know that the Vivax works too. And then we did internal dilutions to see how sensitive we could get it. We got it down to 2.5 parasites per microliter, which is pretty darn good. So somebody was talking yesterday about the necessity to have white papers from inside the target countries you're going to. And I've long-term partnered with African universities. And so we chose the University of Lagos to be our initial clinical site. We dealt with Dr. Oyibo, who is widely published in malaria, and had all the samples that we needed. So his techs were fabulous. They came out, trained with us. We sent a team in there, and we did the installation, and they ran samples. And I don't have all the data from it because it's just recently happened, but initial data looks good. These are the controls that we gave them, and they nailed all the controls without any problem. We then did positive samples, Falciparum. We did adults and children, and we did negative samples just for QC control, and everything came in. The Parasitemia assay has 2 different levels, but it essentially reports out the percentage of infective RBCs in the sample. And we basically, it's an inexpensive stain that we use, so it's a very inexpensive test. It competes with the 55 pence area that we were discussing. But a simple process, you add the stain, you add the buffer, and you add the blood, and you go. And it'll tell you if you have hyperparasitemia. You'll know the level of infection, and for research, you'll know if the drugs are working. And here's an example of running chloroquine over time on it. You can see that the incidence level goes down, and the drug had an impact. So if you're testing new drugs, it's a simple device to use. The parasite density method differs in that we add a lyse to the process, and it reports out the number of parasites per microliter of blood. It's easy to use, it's fast, and it's inexpensive. So this is the team I'm fortunate enough to work with. I consider myself lucky. They're all researchers and scientists. I just am a commercialization expert. Any questions?